A very good evening to everybody, and a warm welcome to all our audience and participants, students, faculty members, and everybody who is connected to AOZ today through this alumni series, episode programs. So I'm glad to present yet another episode, episode number 7 in this series of success stories of alumni to all of you. And very glad to bring yet another zoologist today on our platform. So, those who are connecting with us for the first time, let me give you a brief introduction about what AOZ is exactly about. So, AOZ is basically Association of Zoologists. It's a non-profit organization that provides an immense platform for zoologists across India. We have just been into this, as an organization past 3 years, and we want to do a lot of activities, collaborative research work, training programs, conferences, symposias, and a lot of programs which are relevant to the subject zoology and which provides platform to the young minds to work in the field of zoology. So very soon we are going to come up in the next week, August itself, with another episode of our alumni series. And soon we are starting a lot of offline programs. You can check our programs onto our different social media accounts. And if you are interested in becoming the members, you can write to us at the email which is flashing right now onto your screen: zoologist association 2020 @gmail.com. We are available on Facebook as well as Instagram, and you can contact us through that. And we would be glad enough to collaborate and organize programs with lots of other agencies, universities, and different kinds of institutes and organizations. So, in today's series, let's welcome our today's guest. And before I actually do it, this series is all about those people who are no they are zoologists, and we have especially been organizing this monthly program for the young minds to get inspired to listen to the success stories of these particular people who have utilized zoology somewhere in their life to make a achievable landmark into their careers. So presenting you one such person and one such zoologist today, Dr. Alpesh Patel. Hello sir, good evening.
Yeah. Good evening. Good evening.
So, Dr. Alpesh Patel is based at Ahmedabad presently. He is a geneticist, and he is engaged in a number of institutes with respect to giving his advice and his genetic background for solving different issues related to thalassemia, sickle cell anemia, and genetic disorders. So he is a director and he's also a consultant. He is working basically with Gene Explore Diagnostic and Research Center, Ahmedabad. But he is also linked up with number of hospitals like Genetic Laboratory of Department of Anatomy, BJ Medical College, Ahmedabad. And I'm very glad, sir, to have you today onto our platform and announcing that you work for Indian Red Cross Society Genetic Lab also in Ahmedabad. I'm a very proud life member of Red Cross, and I work for Red Cross as well. So very good to have you, and I cannot stop telling people that he works for Red Cross. So it's a different charm
Thank
to note this, that you work with such an old organization. So it's more than 100 years now for Red Cross to work in this area of sickle cell anemia and thalassemia. So very very proud to have you as in zoologist, at the same time as an part of Red Cross society also. And friends, Dr. Patel is also much into research. So he has done his postdoctorate fellowship again, and he is also into number of research project as an principal investigator. He worked in one of the projects for Gujarat State Biotechnology Mission. He was a co-investor for different projects which were funded by DRDO and CSIR and DST. And he owns so many different kinds of research papers with respect to different aspects of human genetics and cytogenetics. His basic skills are with genetic based diagnostic aspects with reference to more humans, and he is trained into different kinds of microscopy and PCR and plasmid genomic DNA isolation, transformation, primer designing, also for cytotoxicity and genotoxicity assays. So those who aspire to work in this kind of a field, you're going to really have a good time today to know the experiences of Dr. Patel, who is going to share us all lots of things about cell biology and cytogenetics. So, sir, my first question to you would go this way, that most of us usually, when we started the career, BSc chemistry and BSc biochemistry was in limelight, and then came
Okay.
microbiology. So how did you select BSc zoology?
Okay. So first of all, I would like to thank AOZ members to invite me here on the your lecture series. Though it's a success story you have given the name, but I feel it is a success journey that people have to carve a path for themselves. Now coming to your question that how selecting the zoology in the era of where the biochemistry and the chemistry and the engineering like IT are, that time it's really people are picking up those subjects. So it is like when we are after completion of the 12th, people are really not directed. So I have completed my undergraduation from the St. Xavier's College. They give additional 1 year to select your subject, and over the period of time, we thought ki probably the biological sciences are the one which is people are less opting. So there is a higher opportunity in doing something over there. So that is the reason to selecting the zoology basically.
Yeah. So, but like, because it would then usually go that if somebody is into BSc zoology, they'll go to MSc zoology and then PhD zoology. But you also took up a specialization by selecting cytogenetics. So in those
Yeah.
days, even now we have such a good, advanced, technologically sound microscopes available, and such advanced
Mm.
instruments. It is really no easy to work than what it was before. Then how did you selected the specialization that you would wish to
Yes.
work with
So
cytogenetics?
We believe it like zoology and the botany are the major two branches of the life sciences which supports all the other sub branches like biochemistry, microbiology, or if you have now there are lot many courses. I'm not much aware
Yes.
about the too many courses academics how it is working, but at that time there are these are the major two areas where biological sciences people are opting. The zoology and the botany where you at least you get the basic idea about how the your biochemistry will work. For example, very simple example, like if we ask those who have taken the biotechnology at present, if we ask some basic questions of the how the hormone secretions from the different organelles, so they are not that much clear about the concepts. If you have the basic sciences like zoology or microbiology or the botany, so you have the particularly getting the basic your concepts were more clear than the specialized branch. So after completion of the zoology, you have definitely you studied all the biochemistry, you studied all the anatomy of the different animals. That time the dissections are allowed, so we are fortunate to do the lot many systems like vascular systems and reproductive systems. We used to dissect many things like which is still we are thinking it's like the opportunity that we are getting like dissections
Yes.
of the gears all. So those are now not available. So real time experience we are getting. So heartbeats we are seeing like we cannot open the heart and see it, but in the animal systems we have an opportunity at that time that we can do some dissections and understand the anatomy and the physiology of the how the different animal systems, how it is working. So this is more, it carries more interesting interest in that how it is going about. Moving towards after the undergraduation, the towards the post graduation, we are moving towards how the molecules are working in the different aspects. So that is more inclined, more interesting to me. So that's why we have selected the cell biology, in which the specialization subject is the cytogenetics. So we get the opportunity to learn the cytogenetics as well.
Okay, okay. So that's how you came into cytogenetics.
Yeah.
But like, what excites you more in cytogenetics?
So, there are different phenotypes we are observing, like very common phenotypes we can discuss is like Down syndrome, Edwards syndrome, how the chromosomes are playing a role in the phenotype of individual, and how it is changing. That is more exciting. Moreover, there are some cases of the DSD where we have investigated, 46XX male and 46XY female. So phenotype and genotype is different. So how it is working? So that is more exciting to us while we are doing the post graduation. So it is inclining that we need to know that how the chromosomes are, how the cytogenetics are helping them. So after that, picking up the subjects, we come to know not these are only the primary level. So there are lots more, about that. So then we come to know about the cancer cytogenetics, and then the molecular cytogenetics, where we come to know about the unexplored, deletions and duplications associated with the many disorders. Right now, also we are finding very interesting cases where the earlier it was not reported. So these excites me that we need to look it into and exploring, the part of the genetics which is unexplored. That's why we gave the company's name is also Gene Explore Diagnostic and Research Center. So how
Yeah.
this how we are more into the genetics. So how this is done. Yeah.
Yes. So I then have a question that usually, when we are doing experiments for the first time during our graduation or post graduation, the first experience is always a memorable experience. So tell us, like,
Yeah.
when you had seen first time the chromosome, what was your feeling about it?
So actually, I have done the first cytogenetics in the Department of Zoology, Gujarat University. It is a pioneer department for the cytogenetics that we all know. So first chromosome observation opportunity, I got it in I think in the second year of the post graduation, where we have some practical experiments to see the chromosomes and from the different tissues like testes we are doing from the bone marrow of the rat, and then we are doing from human samples as well. So that time when we are seeing, like, it's like how it is coming out, how the chromosomes are, we know we are studying in the theory that prophase metaphase, but actually seeing on the metaphase, then lots of questions we are asking to the teachers. At that time, I think Dr. Devendra Chala is there, Dr. MV Rao is there, and Dr. Divya Chandel, she is taking most of our experiments. So we are actually learning from them. So generally madam is taking the lecture practical, and then we are asking the question, madam, how we are sure that all the chromosomes of one cell will get together only? Why it will not spread? So that's really exciting, the seeing the colorful pictures. So that really exciting that okay. And that time also I remember that the microscope is, it's a undergraduate level, so it's
Yeah.
really difficult to find out the chromosomes under the microscope. And it's not the electronically device, it is, I think, light with the sunlight we are using to see the microscope. So yeah, that memory still I have. So we are using that microscope to see the monocular microscope to see the chromosomes. Yeah.
Yes, yes. So most of the students, they are very impatient to do observations under microscope, and microscopy is like, for many it's a very big challenge. So what would you suggest them how they should develop the inclination towards microscope? Because many a times when they are asked to score the slides, they really want to give up. They are like, नहीं कुछ नहीं दिख रहा है, छोड़ो, अब नहीं देखना है. So
Yeah.
for them, how should they begin to start liking microscope when
So,
they really find it difficult to observe?
so there is, I must say, I remember my guide, Dr. MV Rao. Actually, sir is telling us that my colleague and me that you should not come out from the microscopy room. You have to sit there at least till you are here in the campus. So we have to observe them. In genotoxicity, we need to score the sister chromatid exchanges and the chromosomal operations and all those stuff. So it's really difficult to see it on the microscope for continuously 1 hour or 2 hours. People are not sitting, but it's a pressure, that the guide is, continuously observing and make us on the microscope. Eventually, it's the first 2 weeks, 3 weeks, it little bit we find it torturing, but eventually it is liking. And then I remember that I can sit continuously for 6 hours, like scoring all the experiments together, like 500, 600, 800 plates I can score in one single
Oh,
go. So it is
that's
like
awesome.
you're like sometimes it's coming out of the microscopy room. It's like completely blank because we are continuously seeing on the under the light. Yeah, we also enjoying with the some light music and some fun with the colleagues and all the stuff are going on. But it's like you need to sit on the chair and with the microscope continuously. That will instead of many times what happens that we are looking at the chromosomes. Not we suggested like I have experience like I'm not looking always the chromosomes. I also look the cells, how it look like, how it is, how the if I have treated with some compounds, I will look into the how the cells also has some impact or not. There are some cells which are has a different morphology. So many things. You don't only look on the chromosome, so you learn more thing under the microscope. So that's what currently I'm telling to my students as well, and those who are working with me currently in the as an employee. So I think I told them that you don't look only on the cells and the chromosomes. You look many things. There are lots of things in the under the microscope that you can observe. How there are sometimes the cells are arrested in such a some stage that you didn't expect it that happens. So that will lead to understanding of the different aspects of the cell division and how the cell biology is working. So only genetics rather than you get the more insights on the cell biology as well. So this how we
Yes,
are
yes.
on the microscope and learning the many things. Yeah,
So those students and people who are aspiring to be a geneticist, you need to use the microscope. There is no substitute for you.
yeah, long
You
hours.
need to
Yeah,
sit for long hours. Yes, you need to sit for long hours, and it's all about observation. You learn through observation. So lot
observation.
of observation leads you to become a better and better experienced cytogeneticist. So wonderful to note that you sit for 6 to 7 hours at a stretch, and you can
Yeah,
score like 800 plates, 900 plates. It's really something
yes,
different to look into. And everybody, sir has already guided more than 15 dissertations, and one PhD student is under him, and he owes lot of research papers, more than 30 research papers in international national journal. So he is into the actual genetic research. So sir, please tell us, like, those who really want to take genetics as in career, from where they should start. Like, as in, you started with zoology, but number of times people don't have a specialization available at masters. So how they should begin their career if they want to be a full-fledged geneticist?
yeah. So I think now the time level changed. That time we don't have the human genetic specialization. Now the few colleges and the few departments are, universities are offering the course in the human genetics. So this is a straight way to going into the human genetics subject, and then later on you pursue the career with the clinical genetics, and this is the straight way. So second way is like there are associated life science subjects like biotechnology, biochemistry. So zoology as well because we have the good cell biology structure in the Gujarat University. So those subjects you can opt for, and then you have to pursue the subject for the clinical genetics. And after coming to the clinical genetics, then there are for being the cytogeneticist, and if you want in the clinical segment. So there are certain two or three institutes, those who are now offering the course in the clinical genetics. There is one in the Vellore, CMC Vellore, and one is in the SCPGI Lucknow. Those are offering 2 years course for the clinical genetics, and you pursue that because the patient counseling in the current era, it's really challenging sometimes, as the field of genetics is expanding. So lot more things are, lot many things are changing rapidly. For example, we are starting before 2 years, very small thing like clinical exome. Now we are reaching to the whole exome. Now going forward, we are sequencing the whole genome as well. So the dynamically, the genetic fields are changing, and like in all the fields, like if it is a research field, or if it's a clinical field, or uh, if you are doing something in the microbiology or somewhere in the biotechnology, I think the understanding of the genetic tools to utilize the how to do the transaction, or the things that you need, the basics of some of the genetics understanding, that will help you, definitely help you out to design your experiments and other stuff.
Okay, so you use number of terms like clinical genetics and human genetics and cytogenetics. So please let our young minds know what exactly a geneticist is supposed to do, and what are the different branches of fields available for them to explore.
So, at present, if we talk about the clinical genetics, so in the clinical genetics, the person who has the experience of more than 5 years after the PhD, after the PhD, they are taking the training for the getting into the with the mentor, they can observing the patients, and then they are deciding that depending upon the phenotype, which genetic test is required. So there is a training for the clinical genetics, and these how after the some experience, do the independent counseling, and the prescribing the or investigating the test for that individual, for the phenotype that manifesting in the individual. So likewise, we have clinics, genetic clinics we are running in the Department of Zoology, Gujarat University. So from where I have started my career, we can say that besides with my PhD, I am doing that, enrolled in the clinic as well. So how the clinical genetics interest will develop from there. So clinical genetics, then understanding after the phenotype, you identify that how it is inheriting and what are the tests are required. So these requires the careful investigation of not only the individual which is presenting or which is in front of you. You have to look it into the complete family of the individual. That is called the pedigree of the family, at least 3 to 4 generations. To take the data, you need to put all the individuals which are under the under either they are inheritable, that is we can call as a carrier for the disease, and then you first test the index case, that is we can call it as a those who are presenting to you which has the symptoms. So you first identify the index case, and then you start the investigation for the starting from the basic karyotyping or the microarray, and then the next generation sequencing as and when required, depending upon the physical characteristics or the phenotypes what we are presenting. And then after the investigating, we offer some tests to their family members, and we also suggest them that how they should and what is the risk for their next progeny, next their family
Yeah.
members, and is the onset of the disease and all the other stuff. So lots of things you need to take in consideration while you are counseling the patient. And we need to also, social also we need to taking care of that. If there is a couple and they have some problem, then we need to both the sides we need to balance that. We need to explain that genetic disorders is not in somebody's will and wish that they can pass on. It is naturally occurring, and we need to explain in that extent of that they should not fault finding in each other. So these are the
Yeah.
things we need to explain to the patient. If it is an
So
individual, those
for
who are
yeah, yeah,
individual they
please,
are
please.
suffering, we need to, yeah, we need to explain them that what is the risk factors associated with that.
Okay, so for becoming a clinical cytogeneticist, you basically need to do the PhD and then have an experience of
Yeah,
dealing with
4 to 5 years
the actual,
experience under the
yeah,
mentor. Yeah.
actual cases. Okay, so once somebody has done their post graduation, graduation, later on, there has to be a research factor with respect to
Yeah.
PhD, and
Yeah.
then you can go and become a part of it. So the previously you used the term cancer cytogenetics. So
Yeah.
if somebody is not interested in clinical cytogenetics but wants to do something with cancer, how they should make their career?
So, cancer cytogenetics is a part of the clinical cytogenetics only. So, clinical cytogenetics is like it's not a separate branch.
Okay.
Cancer cytogenetics, it's a branch of the clinical cytogenetics only, and as it is a more complex, like generally non-cancer cytogenetics, we are observing the aneuploidies and deletions and duplication, which is classically most of the time it is the Mendelian inheritance and very straightforward. But when we are talking about the cancer cytogenetics, any number of the chromosomes can be varied. Like for example, it has a
Okay.
trisomy 8 also, it has a simultaneously it has a some translocations as well. So it will be the each there are more than one type of cell lines as well. So characterize or the identification of the chromosomes is bit challenging in the cancer cytogenetics. That's why you need a much higher experience in the cancer cytogenetics. You need to observe more closely. Moreover, the cancer cytogenetics, the chromosomes are not of that good quality. We are not be able to prepare that good quality
Okay.
because of the cancerous nature of the cells. So
Hmm.
that's why the analysis of the chromosomes are a bit difficult for them for the cancer patients.
Okay.
Yeah, so you
Okay.
need
So
more
that's good to
experience in
know
that.
that. Yeah. So it's more complicated compared to regular clinical cytogenetics.
Right.
Okay,
Yeah.
so that was nice to understand that how one should select the branches when they are no trying to aspire and become a part of this genetic subject. So you also work with Red Cross, and there your work is with reference to diseases like sickle cell anemia and thalassemia. So tell us more about it. Where how genetics is no you are going to use genetics with respect to this, and what's your
Um,
job profile there?
So, Red Cross in 2012, I have joined the Red Cross in 2012, almost 10, 11 years. So, they are struggling with the, beta thal major patients to diagnose for the what are the mutations are there. And generally, the poor patients are coming for the clinic, like from the urban health center. Those who are in the lower socio-economical background, they are visiting the clinic, and they are completely outsourcing the all the tests, and it's really costing very high to them. So they approached me that can you help us in the developing some tests by which we can identify the mutations in the parents, and we can counsel them for the prenatal testing. So we have a program for the thalassemia prevention center. It's a thalassemia prevention center
Okay.
where we recruit the pregnant women in the urban health centers. We first do the HB electrophoresis of the mother. If their HB electrophoresis are coming as a minor, that is HbA2 levels were increased, we
Hmm.
then immediately ask for the husband's blood. If both are minor, then these patients are required prenatal testing. So we are calling the those patients to the clinic. We counsel them that it's a requirement of the prenatal testing, and we take the amniotic fluid and the Korean villus sampling under the sonography guidance. We have the expert doctor, Dr. B.I. Patel is there, who is
Hmm.
doing the Korean villus sampling. The fetal medicine will take the samples, and after taking the samples, we have the arms PCR based protocol where we detect almost 26 mutations which is present on the beta globin gene, which includes the sickle cell as well. That the sixth position is the sickle cell, and after the results are coming, we can if it is a normal, we are allowing them to continue the pregnancy. If it is a major, so there is a urban health centers and the gynecologists are associated with the Red Cross Society, which process further for the termination of the pregnancy like that wise. So approximately last 10 years, we have stopped approximately uh, 1,000 bilateral major pregnancies coming to the society.
Oh,
So most the blood requirement for those patients are every month we need to they require the blood.
yeah.
The BCs are not developing, so we need to continuously
Do
give
white blood
them.
from
So that, yeah,
outside.
so that
Yeah.
will be, reduce for the society. The burden to the society is reduced for that, and that these Red Cross Society we are doing very. I am going as a volunteer there, so it's completely
Oh,
charity. And Red Cross Society also not charging that much to the patient, and
patients.
we are doing this for the cause for the society.
So, friends, you can see you can use your expertise for such kind of projects also once you are picking up the career and having an appropriate place to project your expertise. So good to know, sir, that you are a part of such kind of projects also. So you have constantly been telling me that there is a lot of counseling needed to the parents or the couple who wants to go for these kind of thalassemia babies, and they have a genetic background for that. So tell us some experiences where you have really struggled to undertake this kind of counseling, or when you're in your own lab and you have to do some counseling
Yeah.
related to genetic disorders.
Yeah, so there are genetic counselings. It is bit challenging many a times because, for example, a classical example of we are taking beta thalassemia. So HB electrophoresis tells us that it's a beta thalassemia minor.
Hmm.
Uh, so we are recruiting patients for the prenatal testing. Once we do the analysis of the husband and wife, so there are sequential testing we are doing. Like first we check with the common mutations, which is population has the higher frequency of the mutations. So we do the first common five, which is present in Gujarat population. Then we do the another set of mutations. Then it's stepwise. We are going for the sequencing. At the end of the day, we sequence the complete beta globin gene. So things are like if you
Yeah.
find the mutations and it is correlating with the phenotype, it's very good. Like you can solve the problem, but not the always all the time that you are so lucky that you can get the mutations. Approximately 1 to 2% of the time we are not finding the mutations. That time counseling the patients are really difficult. So we need to tell them that we tried with our best technology which is available, but at present we are not finding the mutation in either both of you or either of you. So if either of you are not diagnosed or identified with the mutation which is caused for the your increased HbA2 level, so we are not sure about the how the developing fetus will what is the fate of the developing fetus will be. So in that case, we still go further and we do the testing for the prenatal testing if the sample is available. If parents are okay with that. Sometimes patients are telling that so why you have taken the samples if you're not sure about to mutation identification and all the stuff. So, but it's really tough to explain them because sometimes
Yes.
if some patients are understanding, they are okay. They said okay, but if they are not understanding, they raise the questions till whatever the level they can extend it, so they can raise the questions. And we need to be very calm when we are talking with the patients. We need to understand being on the patient side that if you are on that situation, what you will be, you are taking the steps. So we because they are really in very trouble position because they don't know what will be going to the fate of the developing baby, and it's a really tough moment that we have to take the decision. But still, we have tried. We can try it better, and at least one mutation if we can identify out of the both the parents, then we can test that one in the fetus, and we can counsel them that either the fetus is major or minor or the major or minor or the normal, that kind of situations are rising. In last 15 years of my experience in genetics, I think two or three times it arrives, where two times fetus is normal, so we don't have the problem. But once it's a beta thalassemia major fetus, but the patient has already opted for the termination, so it's not the consequences with the laboratory and the testing part, as well as to the patient, those who are doing that. So these are the two, three difficulties that we are observing while counseling the patient. There are certain sometimes that we need to be really careful that not always your laboratory results are, you have to cross check your results with the other people as well, those who are
Yes.
in the expert field. So we have the interlaboratory comparison program where we are taking participate. It's not
Hmm.
the that always you do in single handedly. We also share some of the things to the, if I have not be able to do it, I will tell someone to, okay, you can try and let me know what the results are if I have some mistake or not. So that extent we are taking the colleagues or the friends and the seniors, those who are in the field, and try to justify the as much as the possible and as much as we can give the best solution to them. That's the most important part, and that is our most efficient trial to give the best result possible with the available sample and available scenario in hand. So that how we can counsel the patient.
Yeah. Yes. So, friends, if you have questions or you want to have some suggestions, please, you're free to post in the comment box and let us know, and I can pick up your question at the end of the session. So, sir, those students who want to be a part of genetics, what kind of skills they would need when they are actually working, no, and executing themselves as a part of a lab?
Okay, so basic skills are required, like the practical skills. I can tell that most importantly, like understanding
Yeah.
of the how the you know uh, how genetic inheritance are working or something like, so basic understanding of the genetics, like what is the Mendelian inheritance, non-Mendelian inheritance, and other stuff. There are lots of things are in the subject that has to be explored properly. So theoretic part has to be very clear. Coming to the practical portion, they should be thorough with the minimum requirement of the PCR skills and the pipetting skills. The other we are, we can put, we can give the training and we can, take them up for the next level. But basic requirement is that at least minimum things they should understand with that level of PCR and the pipetting stuff. So that much is the required. And after that, most importantly, we consider them that while you are working in the clinical genetics, you have the three things, the three to four parameters we require that candidate should have. Once is the sincerity, it should be sincere, hardworking, and honest. This is the minimum requirement, because besides that, if something is happening in the lab and if somebody is doing, not with the clarity of the minds and some mischief, so it will be really tough to find the troubleshooting when it comes to the reporting time. So these three,
Yeah.
if they have these three basic morals, probably the rest of the things, they can learn. If they don't have these, then
Yeah.
it's really difficult. Yeah,
So, friends, you don't have to worry if you don't know fluorescent microscopy or you're poor in
yeah,
handling karyotyping or understanding. That can be even learned, but you need to have the basic fundamental aspects very clear. And once you
absolutely.
start being a part of the lab, by gaining experience, maybe you learn more and more out of it. So this is like all about their skills. Do you feel that they also need some background related to diseases and disorders when they are working into the laboratories?
Working in the laboratories, if they have, it's really good to, while you are performing something and you knowing it, so it is always a very possibility of less error than the, those who are not knowing and learning, performing. So if you're knowing something and then performing, it's always a better. Like you have told that microscopy, if you're not knowing, that's not a problem. But if you know that if the clinical symptoms of this patient is Down syndrome, like, so I should visualize the trisomy 21 carefully. Sometimes you identify the mosaics where the number of cells are very less. That kind of situation you can easily pick up if you're knowing the background. If you're not knowing the background, probably it is missed. That is a possibility. So your work experience will be more better if you're knowing the background. It's
Yeah.
always better. But if you're not knowing, you need to acquire that.
Okay. And what kind of testings are usually done nowadays, so that no, those kind of procedural aspects, maybe they can prepare themselves with when they are actually doing their studies.
Okay, so at the current clinical genetics, we are doing lot many testing. We are doing starting from the karyotyping. We do the fish. We do the PCR based assays. We do the Sanger based assays. We do the microarray based assays and the NGS based assays, where we do lots of expression based assays. We have the single cell genomics also. We are doing it. So in which all the there is after the if you are coming after the post graduation, so at least you have the basics of, like, if you are want to do the cytogenetics, so at least you have the basics of how the chromosomes are developing, how the microscopy has to be handled, how the how you prepare the chromosomes, basic things. If you are not handled too
Yeah.
many times during your practicals in the post graduation or not in the dissertation work you have not opted, so these are the some of the skills you require. If you are in the molecular genetics, then you need to know that how the extraction steps has to be done, how which are the different types of samples you get it while you are in the clinical setup. Like, you get the all kind of samples, like pus samples, you get the sputum, you get the FFP block, like we are taking the wax block, that kind of tissue you can get bone marrow, you can get product of conception, amniotic fluids, CVS sample, everything you will get. So you need to know that how the, if I have the amniotic fluid, if out of
Hmm.
30,000 samples, can I get the extraction, which math protocol I need to utilize. Though there are
Okay.
seniors,
Yeah.
they can guide them, but if you're knowing it pre-handedly, it will be more helpful to gel with the current staff and working with the laboratory efficiently. So you will get more assignments. If you get the more assignments, you learn more.
Hmm.
So these are vice versa. That
Yeah,
if you have,
definitely. So your experience
you
will be increased then.
so you will
Yeah,
faster.
that's
You will
really good.
generally, yeah. Generally, we have a freshers. We have a starting program from the DNA extraction. Then we'll give the PCR, and then stepwise we check their pipetting skills and all those stuff. They are how many times they are making mistakes and all those stuff, and then they move forward further. So I think it took 1.5 to 2 years to reach till the independent sequencing level or the microarray level.
Okay. So, sir, because you handle so much of human material, like, serum, and you're also going through the amniotic fluid and things like that. So, do students also need some background of bioethics or subjects like biosafety or something like that?
So, in the laboratory, as we are the NMBL accredited laboratory, or those who are the NMBL accredited laboratory, they have the initial induction training. Induction training, they are being told about biomedical safety. There is a
Okay.
biosafety level 1 and 2, where how to handle the infectious samples and all those stuff will be given to them. Training for the biomedical waste, how to discard those infectious samples and how to manage those has to be, as per the protocol of the guideline has to be followed. So
Followed.
that has to
Okay.
be, yeah, that has to be trained. Matlab those that is a part of the responsibility of the laboratory, that they will teach them to at least competence enough to perform basic, at least the personal safety and the
Yeah.
sample safety both.
Yes, yes. And because you have done so many projects, research projects, and such a huge number of research writing in terms of research paper, if somebody is willing to do research in cytogenetics, what are the tips that you would give them from your experience?
So it is like pursuing the career in the cytogenetics, either in the cancer cytogenetics or in the clinical genetics, where you are observing the neurological disorders or something. So identification of the your problem is most important, and where the what is the importance of the cytogenetics in that particular phenotype, or the disease you have taken, that needs to be understood, and that whatever the tools you are using it, that will be available to you. So sometimes we have seen that some of the researchers or students, or PhD students, are coming to us and telling that, so we need to study the autism, and we need to do the chromosomes. So, frankly speaking, we don't have a very strong association with the cytogenetics chromosomes with the autism. So then you need to select the tools like in the molecular genetics. So autism cannot be studied by using the chromosomes. So these are the some of the basic things has to be very well understood. And then once you have selected your problem, then you pursue like how to develop the chromosomes, and if it is a cancer genetics, how to identify the chromosome, how you can increase the quality of the plates, how you can separate the chromosomes, because the cancer chromosomes, it's not that easy to separate it out. So then you learn the band, each band by band, and you know uh, big bricks by bricks in it to add your knowledge, and then you get some concrete results out of that. So these it is not the one day you will learn everything. So it's a gradual process, and you have to be patient. Sometimes it happens that you will 1 or 2 years you will not getting sufficient results, or the get some good results, good outputs. But after once you develop the skills, you will do better.
Yes, yes. So, what kind of presently, what is the scenario about research in cytogenetics? What kind of research topics or areas are coming up for new people to work on?
So at present, the newer technologies are available. Like we have the optical genome mapping. So initially,
Hmm.
this is a technology which can detect the deletion, duplications, inversions, which is approximately it is a 70 kb. So it's a very small portion of the chromosome that which is not being able to detect by the karyotyping, conventional karyotyping, are not being detected by the microarray, which is at present available in the cytogenetics, or not being
Hmm.
captured by the NGS, like higher throughput soft instrument as well. So these are the technologies where which where the unidentified objects, where the unidentified clinical patients are there, that can be subjected to these, and you can utilize more effectively the optical genome mapping. This is a really wonderful tool where we are using it in the cancer cytogenetics as well, where there are many chromosomes having the uh,
Hmm.
translocations and the deletions and duplications. So there is always a human error if I am observing a one karyotype and somebody else is observing a one karyotype. Both have might be a separate results. But if you are using the technology like these, you have the deletion duplication at very crisp breakdown with the breakpoints, and it has a more insights. Sometimes some regions where the drugs development areas are there, where you can identify these are the areas where the some of the tyrosine kinase inhibitors are working properly. So it's a really potential subject where we can do the research out that how the things are working, and more effectively how the chromosomes are embryonically, how it is dividing the cells, and how it is arranging, and how the postnatally is arranging. So both are the very, it's a different lots many things are still not discovered, so there's a lots of scope to explore it. Yeah.
Yeah, and same way, you must have like identified such diseases wherein there is huge scope for cytogenetics to be utilized, but still like people are struggling to do work on it. So can you name few of them for our people who really are keen to do research?
So there are cytogenetics involved in the leukemia, where the complex rearrangement, how it is the what is the prognosis of the patient, that is not clearly bifurcated. So we can do the more cytogenetics, and we can use more advanced tool in that. so we can get the
Okay.
information in these regions, that how it is associated with the cytogenetics, and how it can be explored, how it can be take it for further. So there are, I have given the example of the one AML or CML cases, but you can take other leukemia types of leukemia as well. There where the scope is higher. Secondly, if we talk about the other clinical portion, we have, as I have explained, that autism spectrum disorder. So we
Yes.
cannot do the autism spectrum disorder using the karyotyping, but we can do certainly something which is not associated, not explored using the microarray, chromosomal microarray. So where the resolution is increased. So basically, we need to use the advanced tool to identify the disorders which is not earlier being detected. Like if somebody is told that unexplained mental retardation, everything is normal. Like you have the karyotype normal, you have the sequencing by NGS is normal. Then you can explore the microarray and the OGM like technologies where it is not explored. Okay, there are certain neurological disorders where the involvement of the chromosomes we have seen, like there are Angelman and the Prader-Willi regions, chromosome 15q, where we have not explored much. The secondly, we have the
Okay.
chromosome X, where fragile X regions were there. How it is associated. Recently, we have a discussion on that how the fragile X is associated with the infertility. We don't know. So, these are the regions, and then there are the heterochromatin regions of the 1q, 19, q, 9q, and 16q, where we don't have, I think, 16 and 9, not 19, 1, 9, and 16. These are the three chromosomes. Uh, the heterochromatin regions where we don't know how it is associated with the bad aesthetic history or something, because we are not be able to put the any kind of probes at that region, and we are not be able to explore it. So these are the regions. There are others also, many, but these are the few regions which we can focus and do the find it out, how exploring further, that how it can be associated with the disease or the right.
So, going on to the faculty side, because there is so much to input in education, what would be your suggestion through your experience? Like, what kind of topics are must to be included in a syllabus? Because now we are all in a transitory phase of using and putting the National Education Policy 2020. So, lot of people and lot of institutes are changing their syllabus with that respect. So, okay, what should be incorporated? And it's a must for the students looking to the present work profile that is available in clinical genetics.
I think the it is like it has to be what we suggested. It has to be a separate, complete separate cell. You can design a course for the clinical genetics, the human genetics. Those who are taking, we I have some exposure to the human genetics course where they are included the many stuff. But they can still explore further with the basic techniques. They should be thorough with the basic techniques that I have told earlier as well, like the microscopy, how to prepare the chromosomes, the DNA replication kind of thing. Theoretical also they should be clear. And then the PCR, we don't expect them to be expert on the PCR and the higher instrumentation, the sophisticated instrumentation. They should do everything perfectly. We don't expect them. We expect only they should thorough with the theory, how it is working, how it is functioning, and at least some exposure to do some experiment on PCR and extractions properly, quantifications. People are not able to understand the purity of the DNA or something like that. So these are
Yeah.
basic things, which is I think very much possible with the university and the department level or the institute level, where it's very basic things, and then opting the some of them in the dissertation. So you have the 6 months exposure of doing
Hmm.
this stuff, of at least 20-50 samples. So that much exposure will be sufficient to enter into the genetic laboratory or into the some of the experienced, if you want to pursue the PhD in that, also do some research, understanding the how the things are moving further. So that much requirement is basics, and I think it is doable in the department level.
Yes, yes, very much possible. So, friends, Sir and his laboratory, Gene Explore Diagnostic and Research Private Limited, has already collaborated with our AOZ in the past for number of workshops, and they have also offered testing for our AOZ members at a reduction rate. So I would request on that go, sir, what your lab can offer to people, like just you have offered the way to AOZ. Now, what else you offer to people who wants to join with you?
Yes. So actually, there's a clinical services we have. We are pioneer in the. We have researched out some expression based test, and we have in India we are the only one who is doing the endometrium receptivity assay. So that is the microarray based test that we are offering to the all the IVF going patients. We have the very much sophisticated technology on which we do the single cell biopsy from the embryo, and we do analyze all the chromosomes and reduce the rate of the miscarriages in the patients those who are going the in vitro fertilization. We do study the carrier sequencing of the patient and have those who has the family history of the one or other disorders like the whole exome sequencing and the clinical exome sequencing. We are doing it. With, with these, we are also doing the single based testing like monogenic disorder. If you want to do some SNPs, like one or two SNPs, if you want to analyze. So, two directional single sequencing is the best and very cheaper. And then patients, those who require the karyotyping to be done for the translocations and other stuff, that also we are included our menu where we are expand we are at least at present we are doing almost all the technology starting from the karyotyping till the NGS. It is karyotyping, fish, microarray, NGS, everything is available at present, and we are offering all to the patients and those clinics associated with us. We are also providing our facility. Our facility is open for the research students who are in the university, like they can come with the samples. Okay, whatever the charge they can, they can pay, and they can be there with us with our staff, and they can perform it. They can observe the performance when they are testing their samples. So it is they understand that how the samples are processing, how the
Yes.
single sequencing is being processed. If they have some metagenomics, how the instrument, how they are preparing the library, how they are analyzing on the software. There are bioinformatics tools that is being used. So these are the exposure we are giving to the students. Those they come with their own samples. We are allowing them. Our laboratory is open to all those students, those who are working, who others want to work with us. Culture facility also we are allowing them to work. Observing that if they have the good exposure, they can do their experiment themselves also, but
Yeah.
should have they are shown the competency that they can allow they can work independently, and if their guide is agreeing and everything is perfect, we don't have a problem. So we are open to that level, so we don't have anything to be hidden. It's open, everything is open.
So, friends, take good advantage of Dr. Patel and his laboratory, Gene Explore Diagnostic Research Private Limited at Ahmedabad. You can get your samples tested. You yourself can go and learn how to do the testing. You can also take training, do dissertations, or do research at his laboratory. And if you are looking for concession, be AOZ members and avail the concession by becoming our member and going to their laboratory and doing the testing. So, sir, we are moving towards the end of the no program today, and the last question from my side would be that what would be your key advice to the students from your this long journey and experience of your career? What is a takeaway for the students today?
So I've seen that at present, current generations, people are not sticking to one particular interest. Their interests are changing every day. So finding out that the interest that you most love to do, you should not be tired by doing it. So if you find that interest in pursuing that interest continuously, so it will pay you one day. So definitely, it's not the one day. You should have that interest in your subject or whatever things that you are doing it. So you develop the interest. Don't change today these and second day something different. Don't. I've always told to the student that there is one phase of learning. So you complete the phase of learning and then you enter in the phase of earning. So earning phase is lifelong, but the learning phase is very short. So learn as much as the your phase during your phase of learning and gather the experience, and the rest of the things placed automatically. It will carve your path for your life without any hurdles. So be stick with your objectives. Yeah.
Yeah, that was really wonderful to note. I've just got one question from the sideline, and that is, you have developed your own laboratory as in the form of a business, and now you're a full-fledged a private laboratory. So it's very difficult to take the risk of using your expertise many a times and going into a full-fledged business. So what was your experience about this? Because most of the people would think, I have done PhD and I want to do a job and or be a part of a company, but making a huge setup and then running that setup for such a long period of time with good successful result really demands lot of challenging aspects to be covered. So how has your that experience been, and what would be your advice for those who really want to open up their own laboratories?
Okay. So first of all, like it's a dedication that we have started. It's not right now. We are seeing it's a 15,000 square feet a laboratory where you have all the facilities. It's not always like this. We started with the one single microscope and one single culture room. Those who are associated with me during that time, they are knowing us. That I'm personally working alone sometimes. So gradually it is increasing, but the goal and aim doesn't change. The difficulties are coming in the path. Like sometimes we don't have funds, sometimes we don't have patients. We don't have background of the clinicians, those who are supporting to us. So like we have completed from the basic sciences, not from the medical sciences. So it is really difficult initial days. But as we are doing good things continuously, so it will people will start admiring you, and as you develop gradually, then it comes automatically. Then the things will develop further. So those who want that how to start, I will tell them myself. I'm working initially till morning 9 or 8 to evening 10, 11. I don't know when I'm going home once the completion of the work. It's continuous about 3 to 4 years consecutively. So it is like more regularity, and your consistency is developing yourself rather than it's there is no such tip that you can get success overnight. So
Yes.
for developing some laboratories in the genetics, it's like currently there is a people, we have only very few laboratories in the clinical genetics. We can count on fingers, 20, 25, not more than that. Definitely you will find the competitions. Those people don't like competition, so they will make your path difficult. But if you stay on your path, eventually it will grow. Eventually it will grow, because as the acceptance of the genetics in among the population after the COVID is more, so you will get space, and you develop your niche by yourself. You are not disturbing anyone. You are developing your own niche. You have to develop. You need to do continuously. You have to do the research as well. You need to find new things, and developing doing something different that will be helpful for them. So this is how you can select the path and completing the. If you want to do something, you can do it in the this way. So it's not the overnight you will come to open the. That is also possibility, but if you have the strong background of the finance and expertise, you can collaborate and you can do also. So there are multiple ways to do. So these are the two major things. One is the funding for the basic infrastructure, and second is expertise. So these two are the basic requirement for the genetic laboratory that has to be either acquired or either get supported with the others. So this is how you can develop that.
Yes, and the underlying factor is hardcore hard work.
Yeah, that is
Uh,
always there.
yes,
So you
unless
have,
you don't have that, nothing is going to grow.
yeah, that
Yeah,
is
so,
main.
so good to see, sir, and talk to you and go through your journey of genetics. And I hope that our young minds today who have been with us listening to this talk will get inspired wherever you are, whatever you are doing.
Yeah,
Please continue to do that. Consistent hard work leads to the success and not getting breaked off at times. So please take this message for sure. Even if you don't be a part of genetics, but you need to do anything you do it in a consistent manner and with lot of dedication and hard work without losing hope. So on that note, sir, I would be extremely grateful, and I give a big thank you to you on behalf of our association and all
thank
the
you.
zoology fraternity that you took out such a big 1 hour for us. It was a very short notice. We had
Yes,
been trying to reach you out, and looking to your work profile starting from morning 8 to evening, no number known. So it's really tough for you to devote the time for us, but I'm very glad you spared the time for our young minds and students and faculties to get more inspired. Thank you so much, and we all wish you a very best of luck for your further
yeah,
achievements and good work, and may your lab grow still into more
yes,
square
thank you.
feet, and more success comes onto your door. Thank you so much, sir. Thank you.
Thank you. Thank you.
Thank you.